Hi, my name is Mendelev. I was an Austrian monk known as the father of genetics, thanks to my discoveries and contributions to modern genetics. I was born in 1822 in Austria. In 1840s, I graduated school with honors. In the university, I excelled in subjects like physics and math. I need to know I suffered from depression, which made me take a break from my studies. But I graduated in 1843. In the same year, I joined to the St. Thomas Monastery in Brno. In 1851, I was sent to the University of Vienna at the monastery's expense to continue my studies in the science. I studied physics and botany. In 1853, I returned to the University of Vienna for a teaching position. At this time, I began my experiments. I followed the inheritance of seven traits in pea plants, and each trait had two forms. First, I started defining the pure breeding pea plants that consistently showed one form of the trait after generation of self-pollination. Then I crossed these pure breeding lines of plants and recorded the traits of the hybrid progeny. After that, I found out that all the first generation F1 hybrids look like one of the parent plant. For example, all the progeny of a purple and white flowers cross were purple, not pink as blending would have predicted. However, when I allowed the hybrid plants to self-pollinate, the hidden traits will repair in the second generation, F2 hybrid plants. I wanted to let you know that my seminal work was accomplished using the garden pea, Pisum sabitum, to study inheritance. This species naturally self-fertilizes such that pollen encounters ova within individual flowers. By experimenting with the true bearing pea plant, I was able to avoid the appearance of unexpected traits in offspring that may occur if the plants were not true breeding. The garden pea also grows to maturity within one season, meaning that several generations could be evaluated over a relatively short time. Finally, large quantities of garden peas could be cultivated simultaneously, this allowing me to conclude that my results did not come about simply chance. In this experiment, I performed hybridizations, which involve mating two true-breeding individuals that have different traits. This is done by manually transferring pollen from the anther of a mature pea plant of one variety to the stigma of a separate mature pea plant of the second variety. In plants, pollen carries the male gamete sperm to the stigma, a sticky organ that traps pollen and allows the sperm to move down the pistil to the female gametes ova below. To prevent the pea plant that was receiving pollen from self-fertilizing and confounding the results, I decided to remove all of the anthers from the plant's flower before they had a chance to mature. Plants used in the first generation classes were called P, parental generation 1 plants. I collected the seeds belonging to the pea plants that resulted from each cross and grew them the following season, which were called the F1, first filial generation. Then I collected and grew the seeds from the F1 plants to produce the F2, the second filial generation. All the experiments I made extended beyond the F3 generation, and so on. But it was the ratio of characteristic in PFF 1 and 2 generations that were the most intriguing and became the basis for my postulates as dominant and recessive traits. Dominant traits are those that are inherited unchanged in hybridization. Recessive traits become latent or disappear in the offspring of an hybridization. The recessive trait does, however, repair in the progeny of the hybrid offspring. Lately, I propose that plants possess two copies of the trait for the flower color characteristics, and that each parent transmitted one of its two copies to its offspring, where they came together. Moreover, the physical observation of a dominant trait could mean that the genetic composition of the organism included two dominant versions of the characteristic, or that included one dominant and one recessive version. Employed Gordon P. Breeding to research his concept and also printed effects. In 1865, however, they were not admitted before following his passing descent. Removed statements from Purple Blossom. Transferred pollen out of the statement of Snowy Blossom to a couple of Purple Blossom pollinated carpel developed into bunny planted cereals, rabbit, examined hens. Each of Purple Blossom's multiple generations lead pea production, purple blossoms with white blossoms, true breeding parents, F1 production, each of plants needed purple blossoms. Hybrids F2 production, 705 plants had purple blossom, 224 plants had white blossoms. Radio is equal to 3. Principle of segregation clarifies heritability routine of one attribute plants take out a seed. After pure breed father of mother plants ended up crossbreed. Dominating faculties were consistently found from the progeny, whereas recessive faculties had been hidden before first generation F1. Hybrid vegetation were abundant to self-pollinate. Obtain your course current on these particular specific timelines. It features a summary of our own comprehension of genetics from straight 1865, when German-speaking monk Gregor Mendel very first offered defining with their experiments on pea crops, directly to the discovery of double helix in 1953. Now I'm going to talk about my three laws of hereditary. The law of segregation. In this law, I discovered that there are hereditable items and they come in pairs. The law of independent assortment is what I refer to this is that one of the hereditable items from different traits it sources partly from one another, so that the inheritance of one trait is not dependent of the inheritance of another. And finally, the law of dominance. Remember when I said that there was two types of heritable traits, dominant and recessive? Well, in this law, I explained that an organism with alternate forms of heritable item will have the one that is dominant. What influenced me to investigate about plants and inheritance was the topic of hybridization and breeding. Also, my teacher at the university was Johann Carl Nestle, a scientist interested in hereditary traits and agriculture. To me, Gregor Mendel. Finally, we're going to talk about the Punnett square explaining laws. In order to predict genotypes and phenotypes of the offspring of a particular genetic cross, we use a Punnett square, which owes its name to original Punnett. It makes easier the calculation of the probability of each genotype and phenotypes. There are 6 steps. The number 1 is draw a table with the necessary squares. Number 2, assign letters to different alleles. Number 3, gametes of one parent are on the top of the other ones on the left. Step 4, the genotypes of the descendants are not noted within the squares. And number 5, the number of descendants of each genotype is counted and converted into a fraction.